My name is Raya Turner. My pronouns are she, her, and I'm a student in the Masters of Environmental and Life Sciences program here at Brandon University. And today we're here to talk about the Research Manitoba Studentship Award. The Research Manitoba Master's Studentship Award is a scholarship that's given to master's students across Manitoba every single year. This award has helped me greatly as it allows me to deeply prioritize my research. It allows me to be a full-time student rather than having to worry about other sources of income like finding another job. My lab studies comparative animal physiology, and for this research project, I'm looking into the regulatory effects of a certain gene called Estrogen-Related Receptor Alpha. I've always been drawn to how intricate and dynamic our metabolism is. It takes multiple different factors interacting with each other in order to achieve that energy balance. So that's really what drew me to this research. And in zebrafish specifically, there's a lack of information on how these processes work compared to mammals. So that's one of the reasons that we're interested in how this specific regulator may be controlling these processes. ERRα, or estrogen-related receptor alpha, has been more broadly studied in mammals, and it's referred to as a master regulator as it kind of coordinates multiple different genes that are contributing to the balance of lipid metabolism, carbohydrate metabolism, all these areas that are important to give us energy, allowing us to thrive and ultimately to survive. So we want to use zebrafish as they're an important biomedical model. They're used in various different areas of research. So if we can understand how ERRα functions in zebrafish, if there's conservation of function, we might be able to potentially create a tool where we can more easily study the function of these processes, their occurrence in mammals, as well as potentially use a model to improve therapeutics. So far I've done some microinjections and I've been able to reduce the amount of this specific gene ERRα in the zebrafish embryos. There is some comparable results as to what we would see in mammals. So there seems to be an impact on their development. So we know that this gene must be important for their development, but it doesn't impede their survival. And so we've looked into multiple genes that appear to be coordinated by ERRα in mammals, and we aren't seeing that similar change. So there could be some important differences in the pathways that regulate zebrafish metabolism as compared to mammals. We are seeing one important change, however, in the zebrafish that does mimic that of mammals, and that is that ERRα is actually decreasing another important regulator called PERM1. So we know that this is similar. So in my project, we're looking at multiple levels of study. We're looking at the cellular and molecular level, as well as the whole body functioning of the zebrafish. And it's important that when we're studying the molecular functioning of the fish, that we're not just looking into the gene expression alone, because we know that sometimes gene expression and transcript abundance doesn't always directly correlate to the functioning of those important enzymes and proteins. So we want to make sure that we're getting a full picture, and by looking at both transcript abundance and enzyme activity, we can really begin to understand how metabolism functions in the fish. So we know that ERRα has been implicated in multiple different human metabolic diseases, such as type 2 diabetes and cancer. And so by doing this research on zebrafish, we're answering a few different questions. First, we're examining whether the function of metabolism in zebrafish is similar to their mammal counterparts. And if we do see that there's conserved function of these pathways, we'll have an easier model that we can use to understand how human metabolic disorders, how they function and how they come to be. So we could potentially create gene-edited lines of zebrafish that mimic these disorders, and then we can more easily answer the questions that we seek regarding which therapeutics work best to treat these different disorders. So I've been a student of Dr. Chris Lemoyne for the past few years. I started as an undergraduate student. I did my honors thesis with him, and now, of course, I'm doing my master's. I really enjoy being in his laboratory and doing all the exploratory work that we do. I'm very fascinated by it. We're looking at so many different levels of functioning, which is never boring. Just overall at BU, since coming here as an undergraduate student, I've felt very welcomed. The small class sizes are important to me as I'm a rural student from Wawanesa, Manitoba. So overall, it's been really wonderful. I would say do a little bit of reading on what the different labs are working on here. Definitely, if you're interested in zebrafish, you can always come talk to me, reach out to Dr. Lemoyne, and just come visit the lab to see what type of work we're doing and see if it interests you.